Hey guys, welcome back to another science video this time. Once again, we're going through all of the GCSE science papers, starting with combined, then moving on to triple afterwards. I'm also open to comments suggesting if you want me to go through a topic in particular, but I do try and teach at least a little bit during these videos as well, so I don't just kind of give you the mark scheme because that's kind of pointless. But yeah, without further ado, let's get into it with Bio Paper 1, 2023. First question, it says communicable and non-communicable diseases are major causes of ill health. Which disease is a non-communicable disease? So communicable means that you can spread it, so you can catch it from another person. So something like the common cold, flu. So if we look through here, you can catch gonorrhea and AIDS from other people. Now malaria also spreads through protists, so things like mosquitoes. So cancer is the only thing you can't catch. You can't catch cancer from anything else. Moving on. So, obesity is a risk factor for many non-communicable diseases. Give one non-communicable disease that obesity is a risk factor for. Do not refer to the disease given in part 1. So you can't just say cancer again. Something like diabetes would be I was going to say a good one. It's not a good one. But you can also say coronary heart disease, because again you can't catch that. That's something that's developed in yourself. Or heart disease, something along those lines. National policies are used to help people who are obese to lose weight. One national policy is to reduce the amount of sugar added to food and drink. Suggest one other national policy that can be used to help people lose weight. Well, we live in the UK, so you might be aware that a few years ago they put in a sugar tax, which again encourages people to drink and eat things with less sugar in them. But you can also have education. So just education in schools about the dangers of eating sugar or high-fat foods, high-calorie foods, whatever you want to call it. You can have government advertising. You can also have restrictions on advertising. So, for example, with cigarettes, you may have noticed that you've never seen an advert for cigarettes because it's illegal to advertise cigarettes in the UK. And also on the packaging itself, it has to have those disgusting images of like black lungs and stuff like that. So it's another one. Calorie information on takeaways and things like that on restaurant menus. If you travel around the world, not every country has that. That's something well, it's not unique, but it's something that the UK does in particular. Anything along those lines. BMI is one measure of obesity, and it's calculated by taking someone's mass in kilograms, dividing it by their height squared, and then basically the bigger the BMI, the more overweight or obese you are. A person is 1.64 meters tall and has a mass of 69 kilos. Determine the BMI category for the person. So again, we're just substituting into a formula. So it says you take their mass, which is 69 kilos, and you divide it by their height in meters squared. It's already in meters, so divide by 1.64 squared. And we can kind of work out a value here. 69 divided by 1.64 to the power of 2 gives me 25.7, let's call it. Which, if you notice, is in this region here, which means they are overweight. For 3 marks, I think that's actually quite nice. It's not a bad question at all. Just don't be afraid to substitute into equations. Scientists investigated the effect of smoking and of BMI on the birth mass of babies. Women's BMI categories were determined before the women became pregnant. Suggest why these categories were determined before the women became pregnant. Well, if we think about what happens when a woman gets pregnant, is of course they're going to increase their body weight because they're growing a human being inside them, right? So if that baby weighs anything, then of course it's going to add to that. But also women do undergo changes which increase their body mass anyway when they're pregnant. So simply put, women gain mass from pregnancy. Or actually I'm going to say gain mass during pregnancy, because obviously it won't last afterwards. Or at least it doesn't have to last afterwards, because of the baby, right? So logically. Then they give us the results between smokers and non-smokers. I would always recommend having a look at the graph first, or the chart or whatever, just to kind of see what the patterns are. So if you notice, if you're a smoker, the number of people in each category is lower. The mean birth mass of the baby in grams is lower consistently in all 4 categories. So again, all 4 categories, smokers have a lower baby mass than non-smokers. So it says here, give 2 conclusions that can be drawn. So, well, what I just said is one of them. Smokers have lower mean baby mass than non-smokers. And what else could we, what other conclusions? Well, is it just that it's less? Well, if you look, there's actually some kind of discrepancies, right? So for example, for the underweight people, it seems to have had a much bigger impact than, let's say, for overweight people. So you can see how the difference is different. So that's a conclusion you can draw. So you can either say it has the biggest impact for underweight women, or it has the least impact for overweight women. Both of those will also give you the marks. So either it had the lowest effect for overweight women because the difference is smallest, or it had the biggest effect for underweight women because the difference is the biggest. Any one of those two would give you the second mark. So this is specifically about measles, which again you do learn about, and you do need to know some specifics about measles. And it says, describe how the measles virus is transferred from person to person. So it's always through coughs and sneezes. And what happens when you cough or sneeze? Well, there are viruses. What? Alright, viruses in the droplets produced, which are inhaled by other people. It's kind of disgusting, isn't it? Which, by the way, is why you need to cover your mouth when you sneeze or cough. You know? And that's also why people get really sick around kids, because they don't tend to cover their mouths, which means all of that snot and those droplets just inhaled by everyone. But anyways, that's enough ranting. Athlete's foot is also a communicable disease. It's caused by a fungus, which is disgusting. The athlete's foot fungus infects the skin on the feet. Apparently 17% of the UK population have athlete's foot, which is disturbing. Here we have the estimated population is 67,961,900. Calculate how many people are estimated to have athlete's foot. We're just taking 17% of that number. Again, use your maths knowledge. I would change that to a multiplier. So literally just 0.17 times that big number. Also, there is on your calculator a percentage button down here. So you can literally just do 17 shift percentage sign and do that. So 6796190. It's like a phone number. 11.5 million people. That's interesting. And that's it. Not bad. And lastly, athlete's foot fungus grows in moist conditions. So just one way a person could reduce their chance of catching athlete's foot. Well, first of all, if it's moist conditions, that means you want to keep it dry. So when might your feet get wet? Well, simply put, you can dry your feet after washing, or just getting wet in general, like being outside, right? Anything like that. But also you can get this fungicide that you can spray on your foot, which obviously kills fungus, fungicide, right? Don't share socks with other people, things like that. And also don't walk around barefoot, which is just good advice anyway, because you can also get verrucas, which are a different type of disease as well, which are painful and difficult to remove. Figure 2 shows an onion cells viewed using a light microscope, and you can kind of see the cell structure here. Here's the apparatus given to the student. You have a microscope, you have a prepared slide of onion cells, so you don't need to talk about how you prepare them, they're just given, and a ruler. Describe how the student could use this to estimate the mean length. Once again, you don't need to talk about preparing the onion slides because it's already done. Okay, so if you write about that, you will lose marks, or at least you won't gain any marks. So, how would you do it given that equipment? Well, first of all, you want to place the ruler on the microscope stage, right? Because you're going to use the ruler to actually measure the length. So you want to have it like alongside here, and you want to make sure that the microscope is focused on the ruler itself. Okay? Cool. What next? Well, what you're going to do is you're going to measure the diameter of the field of view. So again, you're not going to measure an individual cell, because if you look at the shape, look how bendy and weird it is. It's going to be hard to measure the width, right? So instead, you measure from end to end how wide the field of view is. So maybe like 5 centimeters or something along those lines. Then what you're going to do, so then change the focus to the actual cells. So now you're focusing in on the cells, using the same magnification. and you just count the number of cells that fit across the field of view. Cool. Then all you're going to do is you're going to divide that diameter by the number of cells. So let me give you a demonstration really quick. What you do is you measure from here to here the width, and let's say that width is 5 centimeters, right, for argument's sake. It's going to be smaller than that, but whatever. Then all you do is you count from here to here, going all the way along, how many cells you have. So we have 1, 2, 3 cells, and then a half, so say 3.5, and all you do is you take that diameter and you divide it by 3.5. That's the estimated cell width, right, because you've just taken the number and done it. You could technically also do it vertically as well. It won't make any difference. But anything along those lines will work. Now this question here, it was not answered super well, but it says the circulatory system includes the heart, blood vessels, and blood. The heart pumps the blood in a double circulatory system. What is meant by this? Because some people said, oh, the heart pumps to the blood. The heart pumps to the lungs, for example. You need to be a bit more specific. There are a few different things you can do, but bear in mind you need to get 2 marks. So you can get 1 mark for saying something like the blood travels round the heart twice before it goes round, right? So it passes through the heart twice on each circuit. That only gives you 1 mark. Instead, what they're asking for is specifically what is meant by it. So if you ask your teacher, why is it called a double circulatory system, what would they say? Well, they would say that one side of the heart pumps blood to the lungs and the other side of the heart pumps blood to the rest of the body. Now we do need to be a little bit more specific. We need to say which side. So you can say just left and right side, or you can say right ventricle pumps blood to the lungs, left ventricle pumps blood around the body. Something along those lines. But you have to be very, very, very specific when you're talking about it. Now, heart rate is controlled by a group of cells that act as a pacemaker. Where in the heart is the pacemaker found? It is the right atrium. Cool. Which blood vessel carries deoxygenated blood? And there's a reason why they don't just say arteries and veins, because it's not just one or the other. Well, the pulmonary artery carries deoxygenated blood. Now, it might seem weird because arteries don't, when you first learn about arteries, they carry oxygenated blood. But remember, pulmonary means lungs. So this is the blood that's being pumped to the lungs to become oxygenated. Okay? Whereas the pulmonary vein carries the oxygenated blood back to the heart for it to then be pumped around the heart using the coronary artery or the aorta. So yeah, be careful with that. So the structure of a vein is different from the structure of an artery. One difference is that veins have valves, but arteries do not. But why? Well, what's the point of having a valve? It's to prevent blood flowing backwards. Yeah, that's the point, right? So why would a vein need it but not an artery? Well, what's the difference between artery and a vein? Well, an artery carries blood at a much higher pressure, whereas a vein is at a much lower pressure. So simply put, arteries don't need them because the blood is travelling at such high pressures that it's not at risk of flowing backwards. So this is because blood in veins travels at lower pressure. Cool. Now we have to describe 2 structural differences between a vein and an artery, obviously except valves because they've given that one already. Well, a bunch of things you could say. Lumen. The lumen is basically the empty hole. You know, like when you have a straw, the lumen would be the hole, right, if that makes sense. So the lumen is wider in veins. You can also say that it has a thinner layer of muscle as well, because again it doesn't have to withstand higher pressures, or a thinner layer of elastic tissue. That would also be accepted. A leaf is a plant organ. Figure 4 shows tissues in a leaf, so we have all these different tissues and they're labelled A, B, C and D, which means we're probably going to have to say which one's which. Which tissue is the epidermis? The epidermis is the outer layer of the cell, so it's going to be tissue A for this one. Perfect. And explain how the spongy mesophyll is adapted for its function. So again, if we have a look at this is the spongy mesophyll. How is it adapted for its function? And obviously, more importantly, what is the function in and of itself? Well, the function that we're talking about is to allow gas exchange between carbon dioxide and oxygen, because they need to get rid of oxygen and they need to take in carbon dioxide for photosynthesis. So, how is it adapted? Well, if you notice, these cells here and these cells here, they're packed close together, whereas in the spongy mesophyll, there are large gaps. This is to allow gas to fill them so that we can actually have gas exchange. So we have large air spaces, large spaces between cells. And it does say explain, so you have to say why. Why is this important? To allow gases to diffuse or move through the leaf. So they can leaf the cell. Yeah, get it? Anyways, which allows photosynthesis in layer B or tissue B. Actually, no, to or from. To or from cell B. Right? So the idea is that you can have carbon dioxide coming in and oxygen going out at all times, because tissues B is where photosynthesis takes place. Figure 5 shows a pair of guard cells around a stoma. Cool. During the day, glucose is made in the guard cells. Describe how an increase in glucose concentration in the guard cells causes the stoma to open. Well, did I miss a question? Oh, whoops, missed a question. The xylem is adapted to transport water through a plant. Explain one way xylem is adapted for its function. Well, first of all, it's composed of hollow tubes. Why do they have to be hollow? Well, to allow water to pass through, right? Again, think of it like a giant straw. If your straw had stuff inside it, water's not going to be able to pass through the straw or any liquid. So it's composed of hollow tubes to not obstruct water flow. It does just say explain one method, one way that it's adapted, but some others is it has lignin to keep it open, again, for the same reason with the straw. That's why paper straws suck, because they don't stay open, right? It has no end walls to allow water to flow easily as well. Now onto the question I was talking about. So how an increase in glucose concentration in the guard cell causes the stoma to open. So first of all is, how does the concentration change? Well, water enters the guard cell. Via osmosis. Cool. And what happens here is it causes the cell walls to swell and curve at the same time, which opens the stoma. So if you'd like, imagine you're trying to squeeze these. Like if you pinch them slightly, you can imagine that the hole will get bigger, right? The stoma will get bigger. That's essentially what happens. So the cells swell and curve to open stoma. Stoma. So when the water enters the guard cell, that lowers the glucose concentration. Light intensity varies during 24 hours. Shocking. Figure 6 shows the overall exchange of carbon dioxide and oxygen for a tree at 3 different light intensities. So bright light, you have CO2 coming in, oxygen going out. Dim light, it looks like nothing's happening, but there is something happening, but I'm not going to explain it just yet. And no light is where oxygen comes in and carbon dioxide goes out. So explain these different things. So since it's 6 marks, I'm probably going to put 2 for each of these, okay? So let's get started, shall we? So let's start with bright light. Let's just go right to left. Right to left? Left to right. Good lord. So with bright light, what happens? Well, as you can see, the overall CO2 is coming in and oxygen is leaving. So overall, CO2 enters the tree and oxygen just write oxygen, leaves the tree. This is because at high light intensities, the rate of photosynthesis is much higher than the rate of respiration. So again, when you're thinking about these kinds of gas exchanges, always think about what process is. So what process takes in CO2 and produces oxygen? It's photosynthesis. And here, what process takes in oxygen and releases CO2? It's respiration. So on this side, we have photosynthesis dominant. It's at a much higher rate than respiration, whereas in no light, respiration is dominant. So now, what do you think it is in dim light? Well, it's not, they're balanced. So the rate of photosynthesis and the rate of respiration is basically equal, which means there's no overall change in oxygen or CO2, because every bit of oxygen that goes in is changed into CO2, and every bit of CO2 that comes in is changed into O2. So you have this balance. So the rate of carbon dioxide forming is the same as the rate of oxygen forming. And when I say the same, it's virtually the same. Whereas here, it's photosynthesis is dominant. So we're producing a lot more oxygen than we are using. So, continue with that, I basically just explained the rest of the question as well. So dim light, overall, there is no change in gas exchange. This is because the rate of photosynthesis is equal to the rate of respiration. So in other words, oxygen is used up just as fast as it's produced, and carbon dioxide is used up just as fast as it's produced. And then with no light, when we think about it, there's going to be no photosynthesis. So overall, CO2 leaves the tree and oxygen enters the tree. This is because the rate of respiration is far higher than the rate of photosynthesis. There we go. Perfect. For 6 marks. Not really that bad of a question. Amylase is an enzyme that digests starch in the digestive system. Why does starch have to be digested? Well, it might help to think about what it's digested into. So starch is broken down into glucose and other sugars, but glucose. Why is that better? Well, the reason is because glucose is a much smaller molecule and it can dissolve. So it can actually pass into your blood, which is where we need it to be. Whereas starch is large and insoluble. So starch, large and insoluble. And so must be broken down. Actually no, so cannot be absorbed into blood. Right? Again, that's why I break it down to glucose, which is soluble. Oh boy, this looks like a lot. Uses a colorimeter to investigate the rate of starch digestion. It measures the percentage of light passing through a liquid. The darker the colour of liquid, the less light passes through. So the logic here is, if you think about it, starch can't dissolve, whereas glucose can. Which means, initially, you won't be able, not much light will pass through the liquid because there's lots of starch in it. Starch doesn't dissolve, so it doesn't break apart. But then as it's turned into glucose, you're going to get more light passing through it, okay? So again, concentration of starch increasing, percentage of light passing through decreases. Makes sense. Yep. Suggest what liquid was used for the test with 0 grams per decimeter cubed of starch concentration. So again, actually I didn't read the whole thing. So they mix 1 centimeter cubed of starch with 10 centimeters cubed of iodine, and then measures the part percentage. Okay, cool. So then it'd probably be the initial amount, right? Yeah, for the 0 grams per decimeter cubed, it was probably 1 centimeter cubed of starch with, sorry, not starch, water. So 1 centimeter cubed water. Do we need to write the iodine? Yeah, probably. And 10 centimeters cubed. Was it 10 centimeters cubed? Yes, of iodine. So again, in order to have 0 starch concentration, it means there was no starch, which means they probably just used water. Point 3. Explain the change in the percentage of light passing through different concentrations of starch suspension in iodine solution. So I basically already did this, but let's go through it. So if we look, it's like this exponential decrease. So it decreases extremely quickly for small increases, and then it slowly levels off. So we need to explain why that is. Well, first of all, the percentage of light passing through the liquid decreased with increasing starch concentration. So don't forget, when it says explain, you also need to state what the change is in the first place. Now, why is this? Well, they've mixed iodine. What colour does iodine turn when you mix starch with it? Well, it turns into like a dark purpley colour, right? Or black colour. So, simply put, the more starch concentration, the darker the liquid. So because the iodine solution was darker in colour, and what happened? And if you remember your physics, anything dark will absorb more light. Right, that's why if you paint something black, it gets very hot, and you paint something white, it doesn't get as hot, because black absorbs lots of light, white doesn't. The student then investigated the rate of starch digestion using amylase. This is the method, so they put 10 centimeters cubed of starch suspension into a test tube, put 5 centimeters cubed of amylase solution into a separate test tube. They put both the test tubes into a water bath at 37 degrees for 10 minutes. Then they mix the content. So again, for those 10 minutes, nothing's happening. Well, the experiment hasn't started yet, let's put it that way. Then they put the beaker into the water bath, remove a centimeter cubed of liquid, and add it to iodine solution, measure percentage of light, so on. So the question is, why were they kept in the water bath for 10 minutes before being mixed together? What effect would that have? So what would happen if you put the test tubes into the water bath for 10 minutes? Well, initially, the test tubes are not at 37 degrees. They're at room temperature, which might be around 20 degrees. So when you put them into the water bath, they will start to heat up until they get to 37 degrees Celsius. Why is that important? Well, 37 degrees is the optimum temperature for amylase. So it's basically to make it a fair test by ensuring that they're both at the optimum temperature. So, to get them to reach 37 degrees, which technically is enough to get you the mark, but it's so they'll be at the same temperature or at the optimum temperature as well. That's the more technical reason. Figure 8 shows the results. Cool, so it's increasing instead. Fair enough. Concentration of starch at 3 minutes was 0.3 grams per decimeter cubed. Calculate the mean rate of starch digestion for the first 3 minutes using figure 7 and figure 8. Okay, so concentration at 3 minutes was 0.3 grams per decimeter cubed. Let's have a look at 3 minutes. Okay, cool. And then what was the other figure? Figure 7 they wanted us to use, right? What's this one? Oh boy, I love it when I have to scroll back and forth. Actually, wait, what did this one say again? So 0.3 grams per centimeter cubed. So we're probably going to have to use the percentage of light here, and then use that to work out the amount of time that's been in there. Okay, got it. Cool. So the mean rate means that we're going to be dividing by a time, because again, rate always refers to time. So let's see what we'd have to do. Okay, so let's get started with this. So we want to work out the change in concentration and divide it by time. The good news is we can just divide it by 3 because the time is going to be 3. We know that at 3 minutes it ends up at 0.3, so we're going to do minus 0.3. All we need to do is work out the initial amounts now. So if we look at initial amount, let's go to time t equals 0. That is 10%. And then we want to get a concentration from that, which means we need to go all the way back to the first one. And we're going to read off 10% here to work out what concentration would give us that. So if we go along, that looks to be about, so it goes up in 0.2s, right? 6.2, 4, 6, 8, 10. Yeah, okay. So that's 6.6. So we're all the way down here. And we're just doing, this is actually a fairly hard calculation, to be honest. Well, the calculation isn't hard, but the actual process is. So again, if you noticed, I've taken the difference in the concentrations, dividing it by 3, because that's what they've asked me for, right? Which is 2.1 grams per decimeter cubed. that's yeah, that's kind of depressing. That's a bit difficult, not gonna lie. The investigation was carried out at pH 7. Cool. Explain how the results would be different if the investigation was carried out at pH 1. Use figure 8. So we're going to have something from here at some point. Well, if it's at pH 1, what will happen? Well, first of all, the active site changes shape because it's been denatured, right? This means that starch will not be digested. And, well, what does that mean? What would the results be then? Well, if we look back here, initially the percentage of light passing through the mixture was still 10% anyway, so it would just stay at 10%. So it would stay percentage of light remains at 10%. That's what would happen. And don't forget, when you're talking about the results, you're not just talking about the science behind it, because bear in mind, you can't see this. That's not what you see. What we see is what we measure, which is how the percentage of light changes. So that's what you need to keep in mind with those questions. Antibiotics are drugs used to treat bacterial infections. Mutations in bacteria produce new strains. Some strains of bacteria are resistant to antibiotics. Where do the mutations happen in bacterial cells? You can say anything from plasmid, chromosomes, or DNA all work, which is nice. I would recommend just saying DNA or genes, because that's where mutations happen in every single organisms that have DNA, right? So that might be the easiest one to remember. A scientist investigates which antibiotics, A, B, C, D, and E, killed S. aureus bacteria. So they grow it in a petri dish, they cut 5 small discs of paper, they soak them in different antibiotic solutions, they put them into a petri dish, and they keep it at 37 degrees for 24 hours. 37 degrees because that's like the most prolific growth rate for this bacteria. And any blank spots is basically dead bacteria, right? So E and C, they had no effect on this bacteria. But D had a little bit of an effect, then A, and it looks like B has the biggest clear spot, so that means it would be the most effective. The scientists concluded S. aureus is resistant to C and E. Makes sense. Explain the evidence. Well, there's no clear area. What is happening? Area around C and E, which means that there was no bacteria killed. That's why it's antibiotic resistant, right? Because there's no bacteria killed. The scientists later discovered that S. aureus is not resistant to antibiotic E. Suggest how the method was developed and showed where it's not resistant. So what do you think they would have changed? Well, when you think about medicine, it's not just what medicine you take that has an impact, right? It's also the concentration. So for example, you might take a little bit of paracetamol and not feel better, but if you take a higher concentration of paracetamol, you might well feel better, right? That's because different concentrations of medicines also have an impact. So what probably happened is they used a higher concentration of antibiotic E and then observed that there was a clear area around the disc. That's probably what happened. Cool. Broken bones are sometimes repaired using a metal implant. Cool. The area around an implant can become affected with this bacteria. The infection is usually treated with a long-term course of antibiotics. Long-term use of antibiotics has led to the development of antibiotic-resistant bacteria. Different treatments called stems from bone marrow and from fat tissue have microbial properties. A scientist investigated the effect of 4 treatments on the area around infection. Each treatment was injected into the area around the implant. So the 4 treatments were unreactive solution, antibiotic solution, stem cells from fat tissue containing antibiotic. And it was treated on 5 patients when infection had developed. After 7 days, the scientists calculated the ratio of the area of infection over the total area. Bloody hell. So what was the independent variable in the investigation? So what was the thing that the scientists changed? It was the treatment itself. Right? These 2 things would have been control variables, and this here is the dependent variable because it's what you're measuring. So again, dependent variable is what you measure. What's the outcome? The independent variable is what you change. yeah, is what you change. Actually, that's it. Suggest one advantage of using stem cells from fat tissue rather than stem cells from bone marrow. Well, here's the key thing. Where do you get bone marrow from? You get it from inside bones, right? Which means to get bone marrow from anywhere in your body, they need to drill into your bones and extract it, which is painful and it's also difficult, whereas fat is a lot easier to obtain. So, simply put, you can either say it's easier to obtain or less invasive. I'm going to say easier to obtain. You can also say it's quicker as well, because again, you don't have to drill into someone's bones. Stem cells containing antibiotics were produced by growing the cells for 24 hours in a solution containing the antibiotic. How did the antibiotic enter the stem cell from the solution? Active transport, diffusion, osmosis, translocation. Well, let's think about it. Active transport happens when you go against a concentration gradient. So that would mean the amount of antibiotic inside the cell already is bigger than the amount of antibiotic outside the cell. Does that make any sense? No, right? Because in the solution containing the antibiotic, there should be loads of the antibiotic, right? Absolutely loads of it. So it's not that. It's not osmosis because it's not water. Translocation? Not quite. It is diffusion. Why? Because the concentration of antibiotic is lower inside the cell than outside the cell. Classic. Figure 10 shows the results. So unreactive solution and blah, blah, blah. What is the range of results for the treatment with stem cells only? You're literally taking the difference between these two values, right? The biggest possible value and the smallest one. The biggest one, it looks like it's 0.31. And the lowest one is 0.09, not 99. Yeah. Cool. And then lastly, the student looked at the results and concluded injections of stem cells containing antibiotics should be used to treat all implant-related infections. Evaluate his conclusion. So basically, this is where you're saying pros and cons. You're not saying if it's good or bad. You're saying the good side and the bad side, okay? So I can see why he would say that, because if you look at it, it seems to have had the smallest ratio, just even in terms of a range, so that's valid. Although for some, the antibiotic solution might be better, because again, but think about why that might be better or worse, right? Bear in mind, the stem cells, you have to get them from somewhere. That's quite expensive and invasive, whereas just the antibiotic solution should be quite easy. So for some infections, it might be a better idea just to use the antibiotic solution. That's the kind of idea that you're doing, okay? So let's talk about pros and cons. In fact, I don't need that yet. So pros and cons. Pros is treatment had the smallest area of infection and the smallest range of results. That means it's more reliable, right? So again, with the antibiotic solution, look at the range. So sometimes it worked really well, sometimes it barely worked at all. If you're taking a treatment, right, you want it to work 99% of the time, right? Which this one does. It also has no overlap with the unreactive solution, right? So bear in mind, this is what happens if you just let nature take its course, so to speak. So because there's no overlap, it suggests that it always works. So no overlap with unreactive solution, therefore suggests it's always effective. So when I say always effective, it doesn't mean it always works 100%, but it always has an impact. Now let's talk about the cons. Because don't forget, when you're doing evaluation, you need to have a couple of pros, a couple of cons. Just so you're aware, it's 2 of each. If it was a 6-mark question, it'd be 3 of each. Okay? So we're doing 2 pros, 2 cons. I've given you 3 pros. So what about the biggest cons? Well, the first thing is if you look way back in the beginning of this, it says that they only did it with 5 people, which is not exactly a big sample size. So small sample size. 5 people. What else could we say? As I said before, antibiotic solution did have similar results to the stem cells containing antibiotic, and it also has similar results to the stem cells only, so it doesn't mean that it should be used all the time. Similar results to stem cells only. Yeah, and that'll probably be fine. Bear in mind that when you use an antibiotic, you're always going to run the risk of creating antibiotic-resistant bacteria, which is a really, really big issue. So if we can get away with just using stem cells, why not just use stem cells, right? Why risk the... And again, the difference here is between 0.09 and like, what, 0.03? It's a very small difference. So it might be worth just doing the stem cells only. But anyways, that brings us to the end of the paper. If you found it useful, please do consider subscribing. It does help me out a lot. And again, if you want tuition or you have any questions, my email address is in the description and my website is also in the description. But other than that, I'll see you in the next one.